online at least.
Thank you so much. It's a real honor to be here, a real pleasure. And I'm hoping this can be about building bridges between different fields. And coming from a business school, we're not part of a university. We're not affiliated with a medical school per se, but we love to have relationships and build bridges. And as Sophia was mentioning, a lot of my work has been in kind of Bayesian approaches around highly sequential learning in a variety of applications, many of them industrial, discrete event simulation optimization type of things. And so I've had the good pleasure over the last decade or so of trying to think, well, what would that mean in the space of clinical trials? So I'd like to just maybe share a couple of high-level points of view and then describe a stream of work that we've been working on with some great collaborators. And then I'll focus more specifically on precision medicine trials based on some guidance from Sophia. And afterwards, happy to chat about some of the broader health economic implications as we go forward. So let's just get started. And if you have clarifying questions, please do raise them. Let's make sure we're all together through this whole event. Why did I get into this? Luke is trying to get in. So the reason I got into this was I'd done a lot around Bayesian sequential learning in very different applications. And I've been very interested in healthcare for many years, a lot of it around epidemic modeling originally, informing public health decision-making. I said, well, why can't I take some of these ideas from ranking and selection that I've been doing in discrete event simulation space, optimization space, and bring them to clinical trials? I started to look at, well, what does that clinical trial pipeline look like? I'm sure you've all seen a graph like this of preclinical work, clinical work, phase 1, phase 2, phase 3 trials. And just because you're successful at the phase 3 trial doesn't mean you're going to get adopted and people will purchase that particular medicine. This is even a decade old, $1 billion or $2 billion for new molecular entities, according to this particular source. And I started thinking, well, why is it that we measure clinical effectiveness at a phase 3 trial state, for example, and NICE might be interested in incremental cost effectiveness. So it's a different objective function, depending on whether you're phase 3 or here, going to launch, getting market access. As an operations management person, I'm thinking if you have a flow and different actors along that flow have different objective functions, that can lead to suboptimal performance of the entire system. That's what originally got me triggered in this. Can we do better? How are ways of thinking about this? Now, this particular problem isn't just a story about pharmaceuticals. It's about devices, about care processes. How often do I replace surgical instruments for certain procedures to avoid iatrogenic transmission of Creutzfeldt-Jakob disease, for example? That's a project I had the pleasure to work on with Sheffield. So we might want to have other things that are not necessarily patent protected. So we have private funded and publicly funded trials as well. Okay, so a change in the process for managing surgical instruments would be an example of a publicly funded care process. So it's kind of a broader question than just drug development. And a lot of people care about this. So it turns out, about the time when I was getting started interested in this topic, was a very interesting quote from NHS England, highlighting the aim of baking in the assessment and value of real-world cost of research as a fundamental part of projects. So is it worth it? Should I fund you? The National Institutes for Health Research in 2020 said it's keen to see the design, development, and delivery of innovative, efficient, faster, innovative studies to provide evidence. I think you had done some work along these lines for response adaptive trials, Sophia, and found some very interesting things that adaptive trials are not that much more expensive than more typical trials. So that's in the UK. Other folks are interested in these issues as well, although I understand through time, sometimes perspectives change. Certainly pharmaceutical firms are interested in money and health. And venture capitalists who invest in biomedical research innovations are also very interested in this type of a thing. And this area is is flush with people doing great work in that space. So a lot of the work that has looked at trying to reduce the cost, or looking at this cost issue, these huge numbers that I was mentioning, a lot of that work has been around reducing the cost of a clinical trial. Seems to be a reasonable space. Adaptive trials, response adaptive trials, of course, great work being done here. Multi-arm trials, platform trials, a lot of very interesting things to try to reduce the cost of running a trial and getting solutions faster. At the same time, there's been a parallel set of work around expected value of sample information and looking at what's the health economic benefit. So the colleagues that I've been working with at Sheffield for some time, Alan Brennan's group, has been some of the colleagues in York as well, really looking towards, well, what's the expected value if I were to collect additional information? But that's more at the health economic analysis phase, the health technology assessment phase. Laura Flight, who I think has been not unfamiliar to many of you, had done some work with a number of colleagues. And Laura was a postdoc on some other work that we've been doing with Alan Brennan up at Sheffield. She was noting that, at least at that time, there was an interest in bringing health economic analysis together with response adaptive trials, but not a lot had been done at that point in time. So I kind of took up that charge and said, well, let's think about not just cutting costs, but maybe we need, because we'll make a better health economic decision. I'll get more health for the money spent, where money is the total of the research costs and treatment costs for a defined patient population. And maybe we'll try to convert health benefits using, say, a pound per quality adjusted life year or some other similar metrics, so we can have those be commensurate. So when do we stop the trial was the first question we asked in this series of work. I guess, to which arm do we allocate a patient would be a second set of questions. And so we adopted this highly response adaptive Bayesian bandit models, indexes, you'll see will play a framework here. To start out with, we were assuming continuous valued outcomes because we're measuring qualities, we're measuring costs. Probably reasonable, so it's not really a 0-1 outcome type of story here. We're going to assume that we'd like to optimize things, say, for a social healthcare system, as we find in the UK, rather than modeling the incentives of for-profit pharmaceutical companies and social objectives of governments. To start with, we're going to simplify. We'll bring that in later, maybe. And we'll just start with undiscounted rewards to keep mathematically things a little bit simpler. So that launched a sequence of papers that we've been working on. The first was to say, well, how do we balance the cost of a trial, assuming that if I have a larger sample size, it costs more money, and it takes longer for me to make a decision, as opposed to I stop a trial earlier. And when should I stop? When is the expected value of the sample information of having the option to continue the trial outweighed by the observations? So that one came out in JRSSB a few years back. We then said, that's nice theory. How could this fit into the UK system? And so we, with one of the co-authors on the initial paper, Martin Forster, who was at the University of York at the time, had some good colleagues in the York Trials Unit. And so we started to collaborate with them and said, well, let's take some data from some existing trials or some trials that have been run. Let's try to retrofit. What if we had run this trial using the techniques we were espousing in this paper? What would happen? So we were able to test that out. And based on that initial analysis, we were able to actually get some funding from the NIHR to look at how might it apply to a couple more trials and health technology assessments, again, through the York group. And so that's this one. So I'll perhaps chat a little, for those of you who are interested in how this links to health economics, happy to chat in the period of time just after the seminar. And then following that, we said, well, these are just 2-arm trials. Can we think a little bit more broadly? I know many of you are interested in much more sophisticated trials. We said, well, what about highly multi-arm trials? What about dose-finding trials? phase 2, phase 3 dose-finding trials. Today, I'll chat about how to adapt highly multi-arm trials to precision medicine, where we can measure some characteristics of patients, so we try to find what's the best treatment for each of the patients in a population. So that'll be the focus of today. And that one was just accepted to appear in Management Science, a journal in Institute for Operations Research Management Science, which is, it's not the JASA group, not the Royal Society, it's the Operations Research Group. And then I guess also relatively recently, we had publishing a paper accepted to appear in Management Science around, well, what happens if a for-profit firm is selling a drug into a public system and would like more revenue if the health improvement is better? How does that work? Turns out that a lot of the existing work in the literature is focusing on a corporation would be able to take all of the health surplus. So if you add another quality, you get to keep another 30,000 pounds in terms of pricing. That would be a pure value-based, I guess, approach with maybe a Stackelberg leader in game theory for the company. We've taken a Nash bargaining, cooperative bargaining approach, and it turns out we get some very interesting insights, particularly with respect to some of the ways that the UK Cancer Drugs Fund is in trying to encourage innovations for oncology drugs. So I don't think I'll have time to chat too much about that today. And also current work ongoing with my postdoc, Zyla Lee and Shane Henderson over at Cornell. We're looking at if you could be response adaptive, not only for one trial, but across a bunch of trials that an investor might have. It turns out that there can be some value in doing that. Okay, so that's kind of the overall flow. Like today, focus on can the knowledge of covariate structure accelerate learning? I'm not doing this alone. I've had a lot of great colleagues in that sequence of papers. I'd like to thank them all for all their help. A number of funders, and I'm going to give you the usual caveat: views expressed are mine, not those of the funders, necessarily the co-authors, or the organizations that we're affiliated with. And I'm going to show you a bunch of examples. They're illustrative, not necessarily to give medical advice. I'm not a medical doctor. Okay? So here's a tall list. Don't freak out yet. You can freak out later. Precision medicine. So what's the question that motivated us? Give a mathematical model, give a few algorithms to try to solve that. Can we prove anything? I'll give you some computer simulations of some trials that could be used to support precision medicine, in this case, in the context of sepsis treatment. I'm going to try to summarize that. The bottom two are for later, in case anybody wants to talk about those topics. Okay. So usual approach that we often hear about in the press, of course, in medicine is you've got a medical tradition, you've got a treatment. But I'm sure here we all know that many times we have some characteristics of patients that we can take some biomarkers, and then we wanted to find not the best treatment, but the best treatment strategy, which is a function from covariates to treatment. And this has been quite successful in many oncology applications, starting to get to some other areas, not yet in sepsis, perhaps to the same extent as oncology, but we were working with some folks who are dealing with sepsis at the intensive care unit in Amsterdam, AMC Amsterdam, now UMC. And so this is what got us started along this track. Okay. Now, as we started going down this journey, we were seeing two different types of covariates: predictive covariates, prognostic covariates. Predictive covariates, of course, we know are those that might interact with a treatment. So presence of HER2, for example, and Herceptin, okay, good match there. CYP1A2 is a gene for fast caffeine processing. Given that academics are taken to take coffee as input and theorems as output, I'm guessing we have a lot of CYP1A2 represented in the room here today. So this would be an example of caffeine working with people with a particular biomarker. Prognostic covariate, of course, not interacting with any particular treatment, just influencing the overall outcome. And so the predictive covariates, of course, are the ones that are allowing personalized medicine. And the prognostic covariates, of course, are eating up the variance in all of our estimators. Often, these covariates are studied separately. More and more interest, of course, bring them together. One of the things we've seen around some of the biomedical innovations is that a therapy will be targeting a particular biomarker, and therefore there's a hypothesis up front that you may have a predictive biomarker identified. Can some hypothesized structure like that help accelerate the speed of a clinical trial? That's the question here today. Any questions or observations about the question? Good. Too fast, too slow? Okay. So tell me to go faster or slower as we go along. Okay, so covariates in clinical trials. Okay, so often this, again, a rich history, so much that's been done. I'm sure a lot of it I'm not aware of. If you see gaps, please let me know. Subgroup selection would be one use of covariates, figuring out which group is going to be best served by a drug. Use of prognostic covariates for balancing arms in a trial, so response adaptive to try to even out to get similar number of patients in each arm. Or trial design for precision medicine. A lot of amazingly good work done in this space. And so this is the literature. You could add a million more there, I'm sure. Literature I'm probably more versed in historically would be in the simulation optimization or the ranking and selection literatures, contextual bandit literature, where you're trying to learn through time to maximize rewards, and you make a sequence of treatment decisions, and as a function of the treatment and the covariate you might see, some so-called contextual bandit, rich literature there, and then Bayesian sequential learning is an area that I've been doing a lot of work in. What we're trying to do is to link these streams, and what we're trying to do, I guess, here would be to try to work at the intersection of trying to account for the fact that we might have some suspicions about predictive and prognostic covariate structure. We're not able to fully control the arrival stream of patients. In computer simulation experiments, we can decide the covariate set before making a sample. Here, we're going to assume that patients just arrive at the door to the hospital in the order that they can, and we're going to try to admit them to the trial. We're going to focus first on offline rewards, meaning I'm not going to look at the costs and health benefits for patients in trial. I'm going to observe a bunch of patients. I'm going to stop. I'm going to look at all the data, analyze it, and then make a treatment policy recommendation going forward of the sort that NICE might make. Says, for this type of treatment, you do that. For this type of patient, you do this treatment. You select a function. All the health benefits and rewards are for those patients that are treated over the following 10 years following that technology adoption decision. And in many trials, most trials, there's delays in observations. We don't see the data right away. That's also a difference between usual stochastic simulation, where I say I'd like to make a function evaluation. I get to see the data right away before I make the next observation. So it might be like for hepatitis C, I need to wait 12 weeks to look at my viral load counts and make some estimates. So we're trying to link all of these four things together. This suspected predictive and prognostic covariate structure, there have been some other approaches to that. Lasso would be one technique where you assume everything might be predictive and prognostic with respect to everything. Then we use some sort of sparsity analysis, and then we find a subset of things that we think are active. That's not taking advantage of the suspected biomedical structure of the problem. We're trying to use that structure up front, see if that gives us a benefit.
Can I ask a quick?
Yeah, please.
In the decision where you said you're going to stop and say, I make a recommendation, can the recommendation be to continue experimenting?
So here's the thing. In this particular talk with the precision medicine, I'm going to presume, and I'm super glad you asked this question. I'm going to presume that number of patients that I'm going to try to take. I've got 1,000 patients that I can work with. And how do I randomize them as a function of the covariates that I'm allowed to see before they come in? But I'm not going to find an optimal stopping time in this work. For all the other work, I'm going to allow for optimal stopping times. It just proves to be too challenging mathematically so far. Maybe I need to we need to find a grant to look at this together. But the optimal stopping time with this setup is worth it.
Fixed budget kind of.
So fixed budget, and let's learn as quickly as possible. And I'm motivated by this quality adjusted life years. I'm going to assume that all responses here are normally distributed, but they don't have to be qualities. They can be any normally distributed number you want under this framework. It's only when we go to try to optimize the stopping time that we need to ascribe money. All these. Thank you so much for that question. Okay, so what's our math model? So I've got to come up with a bunch of Greek letters and a bunch of calligraphy letters, as usual. So covariates, I'll call XT. So the little teeth patient to, or T is going to reference how many patients have I enrolled in the trial so far. The teeth patient, T1 up to big T, will be enrolled sequentially. I observe the covariates, I assign a treatment, and I observe their outcome after some delay, big delta. An allocation policy will be a mathematical construct, which is a sequence of decisions from covariates to treatments. And that can depend on the data I've seen so far, H, and the covariate of the next patient to be treated. This history only keeps track of data that I've seen, so I know that I've assigned a patient to an arm for the first big delta time units, but I have seen no data. So it keeps track of all of that. What have you seen? What have you assigned? I'm going to assume a linear response function, as we often do. So the expected value of the output will be some linear regression model, including treatment effects, prognostic terms, and predictive terms. And our beliefs around which terms are active will be reflected through this vector C. So it's 1, ith coordinate is 1 if that term is potentially active, doesn't have to be active, but is allowed to be non-zero, and would be 0 if I want to wipe out that term. So I can have a relatively sparse vector C to signal that I think relatively few covariates are actually potentially active. So I'm going to observe use my allocation policy till all the patients are out, big T of them, and then time delta later, because of the delay, I'm going to wait for all the data to come in, and I'm going to pick the treatment strategy, which is a function which maps the best treatment in expectation for any given patient as a function of their covariates.
What's the unit?
I'm sorry?
What's the unit of the terms? Is that value quantity or?
So it can be here. So in all of the other work, I'm assuming that everything is in health money terms. So I've converted health to money. Here, I don't need to do that because I'm not trying to optimize the stopping time. I'm presuming that in line with Sophia's question, some big T is 1,000 patients. I'm going to randomize those 1,000 patients to be most effective to learn as quickly as possible to maximize this reward. So if I have converted them to money, this will be money. Yep. But what I like to do is, because not always do people feel comfortable converting things. So this will maximize whatever that real valued mean is. So if it's the logarithm of viral load reduction, you can use that. So we need, because it's Bayesian, we got to have a prior. I'll talk a little bit more about computing a prior and fitting that with some empirical Bayes method a little bit later. But we're going to use the standard Bayesian conjugate prior machinery. We will have to refer to something called the knowledge state, which would be our posterior mean at any given time, posterior variance about the uncertain performance of those means. That's a covariance matrix. And then the set of treatment assignments that I've made that have not yet been, where the outcomes haven't been observed yet. Okay, so that's kind of the math side of things. Turns out we can solve this using dynamic programming. So we write the reward function as pick the policy that maximizes the expected health value of future treatments, of future patients. This is the what we'll call the implementation decision. We can solve for this using Bellman's equation, saying at each time step, if we know what our current state is, what we would have done at the time step before. So it's kind of a backwards recursion. And this does give us the optimal policy. So we can prove some structural results along these lines. There does exist a Markovian allocation policy, and we can compute it precisely with this if we knew how to compute that. Problem is it's a little bit hard to compute. So sometimes we need some approximations to be able to compute this. So I'll give you a couple of algorithms to help us make this implementable, because we have a cursive dimensionality going on with extremely high dimensional intervals to solve that Bellman equation. So we're going to build on this tool that's been very, very useful in computer simulation. Some similar tools and techniques are used in greedy one-step look-aheads. The basic idea comes under the name knowledge gradient sometimes, comes under the name myopic one-step look-ahead in other cases. And basically, the idea is for each of the different treatments, we're going to compute an index. We'll call it the FEVI index, F for function and EVI for expected value of information. What would be the expected rewards if I were to allocate one more patient, wait for all the data to come in, make the optimal decision, and compare that with the expected value of information or expected value of making a decision now without the benefit of that additional bit of data. So that's going to be our index. So this, by construction, this is optimal. We have a trial with one patient. Most trials have more than one patient, but we can use this iteratively over and over, and it turns out to be reasonably good. Now, this is something you can compute exactly if the delay is 0 and you have a discrete set of covariate values. It can be computed very quickly using some nice techniques that Peter Fraser has developed with his work. What is a little bit harder to do is if you have a non-zero delay, which is often the case in trials, and if some of the covariates are continuous value. And so we can use Monte Carlo to estimate the expectations there, and we will do so. And we'll call that FEVI with dash MC. And then in some cases, we might want to have some additional randomization in a trial. So we might, with some probability epsilon, and 1 minus epsilon, we either randomize or not randomize. I know many of you are using those sorts of techniques already. So we're going to use the highest index with a certain probability and just pick a treatment at random with some probability epsilon. Those would be some example algorithms we might want to compare. So can we prove anything? It's fine to have an algorithm. I like to prove stuff too, if we can. So to prove stuff, turns out that some of these algorithms actually will converge ultimately to an optimal decision. Not all of them do, or maybe some do only in special cases. So we'll use the standard technique in kind of Bayesian bandit type of a story. We'll try to compare the regret associated with making T observations and see how bad are we going to be. So in order to see how bad we're going to be, we have to know, well, what is the value of having omniscient knowledge up front? So that's our oracle. We can write that as the expected value for any given prior distribution. We can write down on the mean rewards. We can write down if somehow the real world was made known to the decision maker right away, what would be the expected value of that be when you look over the randomness of the world. So that's our oracle expected reward. We were able to show that with a theorem that any algorithm, any allocation policy that allocates the treatment to the arm with the highest index with probability bounded away from 0 all time, then it's asymptotically optimal. In other words, you will ultimately learn the correct answer if you're allowed to sample long enough. Now, the usual proof techniques for such a thing often say you need to sample each of the arms infinitely often. That is not sufficient here. Why? Because if we have 2 arms, A and B, 2 types of patients, 1 and 2, I can always assign 1 to A and 2 to B. I'm sampling each infinitely often as long as I have 50-50 mix of patients, but I'll never learn what's the optimal treatment. So a few kind of side steps need to be done in the proof for that to make sure that everything works out, and it does. And corollary of that theorem is that this one-step look-ahead policy is asymptotically optimal. If you randomize a little bit, of course, you're going to be asymptotically optimal with probability epsilon divided by the number of treatment arms. If you have a discrete finite set of arms, everything's going to work out okay. And the Markov, the Monte Carlo estimator of that index is not always optimal. It's optimal if you have a discrete finite set of covariate values and 0 step look ahead. It's a trivial result, that one. But it seemed to work really, really well in practice. And I'll justify that in a little bit. So we can prove some theorems here. So that's a good thing. How does it work numerically? What's the gain? I'll share with you some observations about a trial that we ran, how should I say this? In this European Sepsis Academy, so that was a European Union funded project with hospitals in London, UCL Hospital, the Academic Medical Center, Amsterdam was the lead hospital in Lyon, France, microbiology lab in Switzerland. So it's kind of a whole network. bioMérieux was one of the industrial partners for this project. So to look at diagnosis and treatment of sepsis. And so I'm not a medical doctor at all. So please don't ask me to treat you for sepsis. Ask me to refer you to the people running to that ICU there. But this is a big thing, right? A lot of people die from this, a huge driver of costs in many countries. And so when I was asked to join in as part of this project, I said, I'd like to have a PhD student to work on sequential learning and predictive medicine or in such clinical trial design. They said yes. So we took a bunch of the data that they had around analyzing blood transcriptomic endotypes based on some work that they had previously published, and said, well, if we were to design a trial to confirm a particular observation of whether certain types of patients, say patients with MARS-3, interact in a better or worse, depending on which type of treatment was used, they had some suspicion that this might be going on. What would a trial design look like that would be very efficient? So that was the goal here. So the outcome of interest, our Ys here, will be a change in SOFA score. So SOFA score typically is a number from 0 to 24, 24 being very, very bad, 0 being very, very good. And it's a measure of organ dysfunction. And if you have a jump in that number of at least 2 over a short time period, then and a suspected infection, then that's declared to be sepsis. And then people like to watch what happens to that value over the next 7 days in the intensive care unit. You'd like to lower that number as much as possible. So we want to lower that number as much as possible. We want to have a negative number here. Our covariates will be those of belief. So they took a very large amount of data from blood typing from about 500 or so patients, and Brendan Skikluna and the colleagues in Amsterdam identified through cluster analysis these 4 endotypes. They also had some data around disease severity in general, other comorbidities. And then they were interested in looking at 8 types of treatments: aggressive or non-aggressive hydrocortisone, aggressive or non-aggressive fluids management, tight glucose control, yes or no. So we have really 2 to the 3 types of treatments here. And we were wondering, well, gosh, MARS-3 might be predictive with respect to fluids management. Can that help us in our trial design? We'll assume that each of the different types have some sort of prognostic effect, potentially. And the Apache score certainly has a prognostic effect. And we'll look at 2 different variations. One is we know exactly that MARS-3 and the aggressive fluids has a delta better treatment. And the other one was, let's just generate random realities through the prior distribution. And so we'll talk about analysis using fixed distribution, which means the MARS-3 is exactly a little bit better than the other treatments versus these other random instance setting where we're just generating random realities and we don't know which one is which. You have to articulate a prior distribution. Well, one way you might think of doing that would be to run a short pilot study, use some empirical Bayes method to fit the various correlations across the treatment arms, aggressive fluids with weak cortisol and aggressive fluids with a lot of cortisol, you'd imagine might have a correlated mean reward. And you could try to use empirical Bayes to do that. One of the things that we found very useful here was to manipulate the prior distribution that would emerge from such a fitting by artificially setting to 0 the mean value of the prognostic effects and the intercept effects. But we have to do something different for, we have a coefficient associated with a covariate and a treatment, so those predictive effects. You want to set those artificially high so that the trial will force you to prove that the reward is lower. And that forces more exploration as you go through this process. So this is somewhat similar to some work that Warren Prof did in their book around optimal learning. But it has to be kind of modified for handling for predictive analysis. And we took our distribution of the covariates to be just following empirical distributions roughly for the Mars 1 through Mars 4 from a 2-hospital study. And we had just assumed low, medium, high, so that we have a discrete number of covariates so that we can actually compute these things exactly, just to start with, and then we'll relax that in a moment. That's our experimental setup. For our performance metric, I'm going to look at the expected regret. For those of you who are interested in the bandit world, this is a familiar term. Basically, it's the expected value of the output from the true best and the thing that you selected as the best treatment for a given arm, averaged over the population distribution. We wanted to compare the various variations that we had. There have been some other people in simulation literature working on precision or what they kind of a contextual learning application in discrete event simulation. That's called the covariate optimal computing budget allocation. So we tried to adapt that for this context. We took the response adaptive bias coin. It's inspired by a few people working quite a lot in trials. We looked at a variation on the bias coin of Pocock and Simon, and then just kind of random assignment. Now, the bias coin scheme and the random, these are more about estimating parameters rather than optimizing treatment. So you don't expect them to do necessarily great, but it'd be interesting to see how well they perform here. We also looked at Thompson sampling, which is, let's take whatever arm, and sample from the distribution of what arm is most likely to be the best, and sample from that probability distribution and choose the arm to assign according to the probability that it's best for that patient. CACTUS Thompson sampling by Dan Russo says, let's have a parameter, say, epsilon, call it a half. Well, with probability half, we pick the most probable from Thompson sampling, and with probability half, we resample until we get something other than the best. So this tends to explore a little bit more than Thompson sampling, so in terms of exploration and exploitation. We also did a version of the philosophy that was about here to optimize not the post-trial patient reward, but the reward to the patients in the trial, so the online learning. So we also tested that one. So what did we learn? So on the vertical axis of this graph, we see on a log scale the expected regret, the expected opportunity cost as a function of the sample size. And this is for the randomly generated covariate values. So the random instances, under the assumption that we know the true correct labeling, we just don't know the rewards of each. And it turns out that, surprise, surprise, the thing that you're optimizing for, if you're optimizing for a particular objective function, you win. So it turns out to be the case, despite the myopic approximation here. We see there's two curves here that kind of seem to flatten out, and they've got a non-zero regret. And that's because these are the algorithms that are estimating parameters, and they're not trying to find the optimal treatment. But random assignment is something that's commonly used in biostatistics, but it doesn't work as well here. So the algorithms that are exploring and committing to a treatment seem to be doing rather better. And top 2 Thompson sampling does pretty much the same as what we've got, not significantly different. And then we see the order of what's beating what here.
How many arms are you considering here?
This example, there's 8 arms, aggressive or non-aggressive cortisol, combined with tight or not tight glucose control with aggressive or non-aggressive fluids. So mix and match any of those combinations. So there's 8 for a trial.
Scale on the y-axis, 10 to the power minus 2.
Yes.
Does that tell the difference is small?
So it looks small. Yeah, and this is the feedback we got during the reviewing process as well. It's a very valid point. So suppose if you had 500 patients, you're really on the order of 10 to the minus 3 improvement. Is that really that big of a deal? Okay, well, let's multiply by how many 100,000s of people get sepsis and multiply a death rate of 20% for those who do. And let's look at 20% of hospital budgets. That turns out to be a fair bit of money and a fair bit of lives. So you're right, it doesn't visually look, 10 to the minus 3 doesn't look phenomenally large, but that's useful. Again, this is just illustrative purposes. I'm not saying we found the right way to treat sepsis, but at least based on this data from the hospitals in Amsterdam and Utrecht, seems to be doing something that's useful, or this could be doing something. If we take a fixed instance, these lines tend to go straighter because now we're not averaging over things, so we're not getting mixtures of of curves. We're tending to get a straighter line, and it turns out to be a little bit clearer for any given problem instance where there is really a benefit. This will pick that up. So again, I'm not doing power curves. I'm not doing, we could add that in, I guess, as a Monte Carlo estimator of the power curve. It's something I probably would want to do, but has not been here. We were not able to prove, there was another question?
Sorry, I might missed it. Was the randomization stratified in any way? Was it just equal regardless of covariance?
So the distribution of the covariates? So the distribution of the covariates. So the actual empirical distributions of the different covariates, Mars 1 through Mars 4, that was taken from the empirical study. So it's not exactly 25, 25, 25, 25%. But everybody had at least 15% within each cluster. And then for the Apache score, we just took low, medium, and high. And that was more ad hoc selected. It's probably not unreasonable.
So yeah, I was asking more about the random comparator. Was that?
Oh, the random comparator. That was just, yeah, so it was stratified. So amongst, and by the way, if it's conditional on the covariates, you're uniform 1 through 8. Unconditional on covariates, you're uniform 1 through 8 as well. Yeah, thank you. Okay, so it turns out that if that time horizon goes to infinity, you're going to converge. We have some short run behavior that is also attractive, so that's nice. Not just asymptotically large. Asymptotically, we're all dead. I think it's something said. We were not able to prove the asymptotic consistency for the Monte Carlo version when covariates are continuous valued or when the delay is not 0. Here, we're going to assume the delay is 0. We have a discrete set of covariates, so we can compute all of our algorithms. It turns out that our indices, when you have a small number of Monte Carlo samples, 1, for example, you're going to do much worse. But by the time you get to 20 or 30 Monte Carlo samples to estimate those indices, that actually is almost indistinguishable for this particular case, where we know we can compute the exact answer. So this is giving some comfort. Turns out that if we have the delay in there where we can't compute the optimal anymore without some heavy duty numerical integration, the Monte Carlo seems to do pretty darn well as we increase the delay. And what's the effect of delay? Well, if the delay is a certain number of patients, so 20 patients, 50 patients, if we don't see what patients have been allocated so far, that's the blind, we're going to lose a lot of we have a big regret here. Whereas if we're accounting for the patients that we know that we've already assigned certain treatments to certain types of patients, we actually can do quite well here. So accounting for what are the patients in the pipeline, what are their characteristics and what treatments we've assigned is actually extremely helpful here. And we can also add in randomization of 20% of patients if we feel like we need a little bit more randomization. It works fine. So that's for what in the bandit literature would be, sample and then commit. What if we want to, in a more usual bandit world, we want to account for the outcomes for the patients in the trial and not necessarily after the trial. That's the online reward. So we can write that. It's a relatively straightforward adaptation using some techniques published by Ilya Ryzhov for non-contextual setting without delays. So we've adapted that to account for delays and contextual learning. So if I put an on up there, that means I'm trying to optimize for the patients in the trial, as opposed to without the on, which is I'm trying to optimize my learning during the trial, which might mean some patients might not be getting what's best for them. I'm exploring so that I can better commit later. We also got a feedback, a really good feedback from everyone was, look, you're proposing this as a mechanism that's an alternative to lasso. Why don't you compare this with lasso? So okay, well, let's also do that. And so we chose a covariate-rich labeling and tried to figure out, well, what would lasso have done on a similar experiment as compared to this type of where we have some knowledge of what might be predictive and prognostic. And we update the labeling intermittently, say after 1 sample, 4 samples, 9 samples, 16 samples, all powers of 2. We update our best guess is the lasso labeling. And it turns out for online rewards, some algorithms have very small cumulative regret. So rather than trying to go down, I guess I still want to go down. The more patients I have, the larger the total number of patients I have to be making inappropriate decisions. So I might have a bigger regret. The algorithms that do not optimize for reward of patients in trial, including the original algorithm that I had just proposed, those do quite bad. Because those ones are really trying to either estimate parameters or, like ours, trying to optimize the best decision of a function from covariates to treatment at the end of the day. But as soon as I adapt that thing to start looking at regret of patients in trial, it does really quite well. And so Thompson sampling, which is known to be quite good, is this middle guy. And we're able to get a somewhat better regret, at least for this particular trial that we're just discussing here. I'd love to try it on more trials just to see how broad that is, but it seems to do well. And the top 2 Thompson sampling seems to do a little bit worse. That's expected. Turns out that top 2 Thompson sampling actually works really well for both online rewards and offline rewards, which is very interesting. And there's not a lot of tuning parameters to that. So there's some value to, there's some really good value to that particular approach. But if you really know what you're trying to optimize, you can do a little bit better.
So if I remember well, top 2 Thompson sampling uses a tuning parameter like beta.
Yeah, there's one tuning parameter. It's the probability that you choose the not the best, the most probable.
Do you fix it in advance?
Yeah, you fix that in advance. In this particular example, we used 0.5. And actually in the online companion for the paper that was just accepted in the appendices, we show a couple of different values of that, and we show different ways to mix and match to weight the learning for patients and the learn in trial and the learning for patients post-trial. And so we can look at Pareto curves of what's doing really well. And both are competitive. The thing that I like about the approach we've taken is that if I know there's 1,000 patients in trial, and I'm going to try to make a decision for 1 million patients after, I know those weights, 1,000 and 1 million. So if I can predict, well, what's the expected disease burden for the next 10 years, I can actually have a reason to set those parameter weights. Whereas 0.3, 0.4, I have to tune that typically. I'd rather take something that's observable and use that to tune rather than pick something. The good news is that algorithm works pretty good overall, but we can do a little bit better. Yeah. Okay, so another thing that you might have a concern about is, well, Steve, you said you think you have a suspicion that something might be predictive or something might be prognostic. What if you got it wrong? So if you, let's think through that. If I think that more covariates are potentially predictive with respect to more treatments, I've added more cadence in my regression that I have to estimate. So because I've got more parameters to estimate, you might imagine that inference will be slower. That makes sense. If I put in a little bit fewer covariate interactions than might be the case, I'm missing out on the opportunity to do precision medicine. So I might not ever get the optimal treatment if I put too few covariates in of the predictive sort. And it turns out that would be the case. In this particular example, I'm saying, what if we wrongly assume that all covariates are prognostic and none are predictive? Well, none of them can get that regret to go down to 0 as we increase the sample size. If everything is incorrectly assumed to be predictive, we will ultimately learn it. It's just going to be much slower at getting to the answer. And so these curves are much higher as a result. But this kind of intuition we can quantify, right? The intuition makes sense. We can quantify that. What if we were to try to assume that everything had potentially predictive, and rather than assume we have to fit everything, let's just throw in a lasso once in a while, try to cut the number of covariates we have to estimate, and we'll re-update that every once in a while based on the data we have. And that turns out that that works reasonably well. It doesn't work as well as having the right knowledge of the actual structure, which is the lowest curve, but it's not as bad as just kind of randomly throwing darts at a wall or presuming that you can't do precision medicine. You will actually ultimately learn. We don't have a proof asymptotically that will converge, but it seems to work well in experiments. It's just slower. Yeah, so we had another example. This was our sepsis example from the Academic Medical Center study in Amsterdam. Here we took some other trial data for a 2-arm trial, also in sepsis space, just to see how that would work with lasso, and we got similar results. So we tried it on a couple of different trials here. Basically the same intuition is all there. So lasso can help, but you do lose something. So what am I hoping got said here? So we have some theoretical results here around large sample asymptotics. So that's reassuring. We have some nice computational results that show intermediate term. This is an approach that seems to make sense. And the thing that makes sense is that if you do have contextual knowledge of what might be predictive or prognostic, and the whole point of precision medicine with biotech companies around here is that we think we've got a biomarker that we can interact with. Coffee and SIP1 with theorem proving, right? HER2 and Herceptin for breast cancer, a variety of other applications. So if you have some suspicion, this can actually help quite a bit. And key to doing that was this thing that we can't prove structural results for when you have continuous covariates, but empirically works super well. This Monte Carlo estimator of these indices actually is relatively easy to compute. We've got publicly available code on Andres's GitHub site if you'd like to play with it. It's written in the Julia programming language. And he's working very hard actually to make this a little bit more broadly useful for other people to adopt it. And so we're happy to try to have a dialogue with you if you'd like to give that a try out. One of the key drivers, the insights from the simulation optimization community is to make use of something that colleague Peter Fraser at Cornell calls an H function for the correlated knowledge gradient. We're using that plus some common random numbers to try to test, or as we're making estimates of these indices, we want to have each patient pair see the same pipeline outcomes for pipeline patients as part of that high dimensional integration that we need to compute. And those two tricks make for a very effective index rule. If you don't use that fact, you'll still do reasonably well, but it will not be as good, right? So you will lose some of your learning efficiency without that trick. I'm very grateful for the insights that came out of that original paper. If we don't know the labeling, we can either lose speed of inference, which is not too bad, or we can lose personalized treatment, which is rather worse. And if we don't have that knowledge of the structure, if we'd like some sort of reassurance to validate some of our assumptions, we can throw a lasso step in every once in a while, and that seems to work really well. This is for the learn then commit approach, or learn then earn, sometimes it's called, but the online version of this also works well, or Online Bandit. And we can make combinations. So all those algorithms are available in Andres's GitHub site. Things that we have not done as much would be thinking about other forms of enrichment, predictive or prognostic enrichment. For example, we're just assuming that's the same stream of patients coming in. We might choose to do something a little bit different from that. Maybe we don't want to enroll every patient that comes in if we've got a finite budget. So that would be something that would be very interesting to do. Adaptive stopping times in this precision medicine. We've got the adaptive stopping times for most of the other stuff, but not yet for this precision medicine stuff. I think that would be a very interesting problem to work on. We've made some assumptions, probably should have been a little bit more explicit about that, that we have homoscedastic responses. Looking at heteroscedastic responses, how that might be inferred. Sophia, you've got some great work and others here on 0-1 outcomes. We haven't really looked at that as much. Maybe using some sort of linkage function could be useful in this context. We haven't looked at longitudinal studies. There's been some nice work by some folks at Wharton, University of Michigan, now at Maryland, around surrogate measures through time. Can we use that? So we haven't done that yet, but that could be kind of fun to look at. We haven't looked at value-based pricing for the medicines that might come out of this. This is not a patentable thing to look at aggressive or non-aggressive control of fluids, and the treatments we've talked about are not patentable. But if for a new drug, you might want to talk about value-based pricing. We haven't looked at that yet. So these are some things that are wide open. Lots of fun stuff to look at here. Any questions or observations or? Yeah.